Podcasts you love.
From people you trust.
This is Twit.
This is Triangulation, episode 433, recorded Friday, March 3rd, 2023. The father of modern genomics. Listeners of this program get an ad-free version if they're members of Club Twit. $7 a month gives you ad-free versions of all of our shows, plus membership in the Club Twit Discord, a great clubhouse for Twit listeners. And finally, the Twit Plus feed with shows like Stacy's Book Club, The Untitled Linux Show, The Giz Fizz, and more. Go to twit.tv/clubtwit. And thanks for your support. It's time for Triangulation. Boy, we have a really good one for you this week, folks. I have long been aware of Dr. George Church. In fact, I tried to get into his personal genome project some years ago. He's a professor of genetics at Harvard Medical School, professor of health sciences and technology at Harvard University and MIT. He is often called the father of modern genomics. His pioneering work has contributed to the development of DNA sequencing and genome engineering technologies. He's received many awards, many patents, over 550 publications. A couple of years ago, he wrote a book called Regenesis, How Synthetic Biology Will Reinvent Nature and Ourselves. As I mentioned, he initiated the Personal Genome Project, started over 20 companies. His most recent company, Nebula, offers gene sequencing, full genome sequencing, to you and me for just a few $100s. Let's welcome Dr. George Church. Welcome. It's so nice. It's an honor, frankly, to meet you. Thank you for joining us. Thank you for taking your lunch hour to join us. I appreciate it.
I'm happy to be here.
Although you told me earlier you don't eat lunch, so thank you for. Right. But this might be a time that you use for other things, so I appreciate it. I know you're actually incredibly busy. I read the list of accomplishments in it, and I read the short list. Otherwise, this would be a much longer show. But when you went to school, you studied zoology, not genomics.
Zoology and chemistry.
And chemistry. Right.
Those are my degrees.
How did you move into genomics?
As I was finishing up those degrees as an undergraduate, I did research in X-ray crystallography at the medical school at Duke. And that happened to be the first nucleic acid structure, meaning an RNA, and did a little bit on DNA as well. And I just got infatuated with, I typed in all of the tRNA sequences that were known at the time and folded them up. And I thought this would be a great thing to do for all the DNA in the world, maybe humans. And so then I started looking for a lab to do that in, and Wally Gilbert's lab suggested itself for my graduate school.
Was it the intellectual pleasure of it, or was it that you thought this could make a big difference in the world?
I think it was a little of each. I mean, I was, quite young at the time. I was about 19 or 20, and I think it was more on the intellectual side. But as I got almost as soon as I went to graduate school, I started thinking about applications as well.
And you found quite a few in the subsequent years. I mean, you've founded, 50 companies. Your research is very broad ranging, but would you say, an overarching kind of mission statement or premise to what you're looking at?
There's actually a mission statement on my website. I can't remember what it is exactly, but it was technology development that ranges from basic science to things that are positive for society, and this includes reducing the cost of technology so they can be equitably distributed. Fairly, even the less wealthy nations and individuals can benefit.
In the 40 years you've been studying this, there have been massive changes and shifts, some of which you've been involved in. And when you talk about cost, I've seen your your graph, it beats Moore's law, doesn't it? The cost of sequencing the human genome.
Yes, it's been about a 20 million fold reduction in price and a similar improvement in quality. Over about a little over a decade or decade and a half.
Yeah, going from billions to hundreds, I guess.
Right, from about 3 billion to 300, and at the same time changing the original $3 billion genome was only kind of half a genome. Each of us has contributions from the mother and father. This is as if you just had one of those, or kind of a mismatch of the two. So clinical genomes today really require that you separate out the contributions of your mother and your father.
Yeah.
And that way you can know whether you have two copies or one or zero.
In fact, since then, the human genome was sequenced. And that was kind of a amazing accomplishment in such a short period of time, as I remember. It was a big deal when that happened.
Yeah, well, it's been announced several times, but to a certain extent, it hasn't happened yet.
Oh.
Yeah. So in 2001, it was a draft sequence. 2004, it was a so-called complete sequence, which means 93%. Just this past year, it was 100% complete, but for only one genome. Again, not both your mother and your father.
Yeah.
And so there is not yet a complete genome for any human being, any real person. And even once that's done, I still won't consider a victory until we have it at a cost that everybody can afford.
Right.
So, right now we're around 95% at a price that probably everybody could afford if the healthcare system, can get probably a tenfold return on investment and provide it for free to everybody.
Is that why you've started so many companies? You're really quite a serial entrepreneur. Is it with that goal to kind of, by turning it into a company, making it financially stable?
Yeah, it's almost impossible to do this directly from academia.
Okay.
Even if you license it out to a company, they're not likely to pull it off because they don't have the know-how or the motivation.
The incentives are different too, right?
Incentives, yeah. And so, but it's hard to scale it and market it and do customer service and all that stuff inside academia. So you really have to have a transition between the two that typically involves some of the people that invented it, like the postdoctoral fellows from my lab, have to accompany it out as kind of a package deal and get it going.
Yeah.
So I've tried other ways, and it's really, this way works.
Good.
Gets things out there.
Yeah. Why do we want to sequence the human genome or any genome? They've sequenced what, Drosophila, some fruit flies, something, right? We fully sequenced.
The only thing that I think is fully sequenced that's an animal is a nematode.
Okay.
It's a very small genome.
And what do we learn from that?
I don't think we need to sequence the full genome to get most of the societal benefits. But I think we do need to do human genomes, and lots of them. And we're doing that now. We're doing hundreds of thousands of human genomes have been sequenced and made available to the public. And this allows us to do, I think the most important thing that for now anyway, is it allows us to avoid very serious genetic diseases that are typically called recessive, and they're done by carrier screening. And so this can be done, this could save us $1 trillion a year if fully implemented, but it already saves a lot of very serious diseases that kill, for example, kill children before they're 6 years old, or so that's the main thing. And then there are other things that can be done that involve developing new drugs, developing gene therapies, in dealing with the age-related diseases. Those all are greatly helped by human genomes, and then infectious agents. We can get those done in record time now. because of the same kind of technology.
Is that what the mRNA process that coming up with the COVID vaccine involved, was sequencing the COVID?
Right. Well, So being able to sequence the coronavirus genome very fast. In principle, if you have a new pathogen, you can get it sequenced out to the public, to the scientists who need it, within a few days.
Yeah, it was over a weekend, I think, as I remember.
Yeah, and then you can go to a prototype vaccine quite quickly. It doesn't have to be messenger RNA; it can also be DNA as well. But essentially, all of the top vaccines, about 5 out of the top 6, were formulated as gene therapies. And a gene therapy is you just write you take the DNA that you just read and put it into a vector, either a lipid vector or a viral protein vector, to get it into the, say, intramuscular injection is typical. And so that's becoming routine, but it's amazing how quick it wasn't routine 3 years ago. And in fact, two new gene therapy methods were incorporated into the COVID vaccines and approved within 11 months.
It's remarkable. Yeah.
Some of them anyway. Yeah.
Yeah.
Mm-hmm.
Is this it's a revolution then in how vaccines can be developed?
I think the ability to go from DNA sequence to DNA or RNA vaccines is really a revolution.
Yeah.
And in fact, you can see vaccines were kind of a sleepy technology before COVID, and now they're making vaccines to norovirus and rhinovirus and basically pneumonia and sometimes 20 different strains, Lyme disease. It's gotten a lot of excitement.
Every time I go to the doctor, I get a new shot. And happily, by the way, I'd much rather have the shot than the shingles.
Yeah, it's funny. We get inoculated to random things every day, where we get a cut or eat something, breathe in. We're getting all kinds of random vaccinations. I would prefer to have non-random ones. So. Maybe both.
A little bit of both. Yeah. Don't eat dirt, but you don't stay away from it either. So, boy, there's so many areas I want to talk to you about. You said something interesting, gene therapy. So obviously, the initial use you test the parents to see if they have sickle cell anemia, and then you can, there's nothing you could do about it, I guess, but you can at least counsel them, right, about the presence of that gene.
You can do it both ways. There now are gene therapies for sickle cell, and you can do counseling, which is considerably less expensive. So hemoglobinopathies in general, which includes sickle cell, can be up to $3.5 million for gene therapy, but they can be as little as $10 for a genetic test, and maybe another $100 for the counseling.
The phrase gene therapy sounds very science fiction. What is gene therapy? What does that involve?
Well, it really is just usually one gene, but it can be many, delivered to human cells. Usually it can be intramuscular, intravenous, and that gene then either replaces something that's missing, or it provides you with an antigen that stimulates your immune system. That's what vaccines do. And like I said, vaccines are increasingly formulated as gene therapies.
Right.
So it's just a gene or one or more genes put in a form where they will get into a particular cell. It could be targeted for the cells that you want to impact most.
Okay, so and then I was thinking more like CRISPR, like cutting and pasting genes and chopping them up. We're starting to be able to do that as well.
Yes and no. I would say first a little appreciated fact, just like it's a little appreciated we haven't finished the human genome yet. A little appreciated fact is that 100% of all of the gene therapies that are FDA approved right now, which is 16 different gene therapies, none of them involve CRISPR.
Right.
So, and now that'll change. It'll be 90% soon, but but I think it's likely that it will remain. Most gene therapies will probably not involve CRISPR, partly because they're pre-CRISPR and post-CRISPR technologies that are moving at least as fast.
Oh, interesting. Okay.
Yeah.
So, yeah, to some degree, I kind of got the impression, again, as a complete layman, that CRISPR was a little bit of a disappointment. Is that the case?
I think it's good for research. There was a lot of hype,
Right.
and I have to say I've been the anti-hype, even though I was, benefited financially from it. I tried to keep people thinking about things, alternatives, because my experience with sequencing is there were always alternatives, and they just kept replacing previous ones, sometimes with almost no continuity. Like there's almost no relationship between nanopore sequencing and fluorescent sequencing. And then, but it has definitely stimulated interest. It was a catchy term or something. I don't know exactly why I think it was just people hadn't been paying attention.
Right.
And there's this revolution, which was really sequencing and synthesis and delivery, kind of overtook them, and they just gave it a name, CRISPR, which really doesn't apply to any of those three necessarily.
That's how it is in this business. And I'm talking about the journalism business. We dimly understand what you're doing, and so if we can grab onto something,
well, that's the scientist's fault.
No, I
they
disagree.
Don't communicate it. No, it is.
It's not an easy thing to do.
At it.
Yeah. Well, but because you're dealing with such complicated subjects, you're very good, though. I have to say, you're very good at explaining this so that we lay people can understand it, and it's a very exciting. I think a lot of people think that just as the computer revolution really changed our world, the information revolution, that the genomics, the synthetic biology revolution is going to change the world. In fact, I've been hearing that for decades now.
Right. You're seeing it. You're hearing it and seeing it in the marketplace.
It's happening. Yeah. It also raises ethical questions that we're not well equipped to deal with. If you can, decide to have more boys than girls, or you can decide you want your boys to be smarter, or all kinds of things. Are we getting closer to that frame? And
well, certainly boys versus girls is not synthetic biology.
It's actually easy.
It's easy via in vitro fertilization.
Right,
so that's reproductive technology that's dates back to the.
But the idea of making superhumans, perhaps.
Super, yeah. So that probably is inevitable in a certain sense, in the sense that we're trying to cure diseases, and some of the side effects of curing diseases is you create things that help people that aren't sick. So in other words, if you try to reduce cognitive decline, let's say in your you're 90 years old, and then you apply that to somebody who's 50 and is not in cognitive decline, it might actually cause cognitive enhancement.
Right.
And there's a lot of examples of cognitive enhancement in animal models. They might be very specific for a small number of tests, but it doesn't exclude the possibility they could hit the kind of cognitive that people want. So
it was, in fact, wasn't that the Chinese scientist who modified the DNA to increase resistance to disease?
Right. So J.K. He was in prison for 3 years for making one child, possibly engineering three, but making one of them plausibly resistant to HIV by changing one gene called CCR5. That was already being done in adults that had HIV, and it showed some promise. And there are people walking around that have it naturally that weren't genetically modified that are HIV resistant. So that's plausible, but you don't need to engineer the germline, which is what he got in trouble for. Almost everything that you can do with gene therapies in germline or reproductive, changing the inheritance, you can do after they're born. The only exceptions are things that happen very early in life, and those I think fall in the category where genetic counseling will work.
Right.
I think and many of these things can be done without any biology at all. I mean, we're greatly enhanced physically, due to jets and forklifts and things better smartphones. So you need to focus in on the things that you actually can uniquely do with gene therapy, and most of those you can do in adults.
Would you avoid germline therapy, or is that something down the road that you think is?
Well, it's kind of asking if I would avoid jetpacks. I just don't need a jetpack, frankly.
Good.
In fact, at this point, I don't really even need jets, because I could go at the speed of light talking to you.
That's right. That's right. We didn't have to meet in the middle.
Even though we're three time zones apart.
Yeah. So you've done some work on aging as well. And actually, that raises ethical issues, because what if we could double the human lifespan? Should we? Yeah.
So I think there are, reasons that come up for why you might not want to, and then there are counter arguments. I think one of them is population explosion. That would we're already on our way to population stabilization and maybe even implosion, because most people are living in cities now, and that fraction is increasing. And when they live in cities, there's a tendency to drop family size expectations from around 7 children per family down to 1.2. And it requires 2.1.
That's negative growth. Yeah.
Just to get replacement. So 1.2 is negative. So you could argue that we maybe need something to hit a balance there. Also, there's some talk that, once we eliminate poverty, then we should probably get off the planet, because we're sitting ducks on the planet. And that also would cause a drain.
Sure.
So that's population. And then there's the possibility that older people would be rigid and not change their mind, and then not be flexible. But of course, that's based on the model of current old people, and it's not even true for current old people, because I know a lot of them that are more flexible in their views than some knee-jerk 20-year-olds.
So, setting aside the concerns of the ethical concerns, can we extend life? How close are we to doing that?
So we have done it in animals by a number of different ways. We have also done aging reversal, which is an easier thing to prove. So, since the variation in human lifespan is already, multi-decades,
right?
Where some people die of age-related causes when they're in their 60s, and other people make it to 110 or more. So to prove that you've actually extended somebody's life might require a pretty big clinical trial that lasts for 30 years, which is...
By then I'll be dead, so that's no good. We got to work on it.
Also, whoever funded it will be broke. So instead, what you do is you do a reversal of age-related diseases, which, by the way, is also easier to get FDA approval for.
Right.
And then that same drug, if you're getting at the core of aging, in order just to deal with, cardiovascular disease, then you might that same drug might also work for neurodegenerative diseases and maybe even infectious diseases. And so those are almost everything has an age-related component to it. So I think the answer is, we're making progress. At least 2 recent gene therapies, which is my favorite way of reversing aging or dealing with age-related diseases. At least 2 of those have been shown to have the side benefit in animals of causing a shift in average longevity.
Right.
Even though that isn't necessarily how they're going to get approved by the FDA. It's already been shown in peer-reviewed papers.
Do we know why we age? Do we know what the causes of aging are?
We know that, so there's 9 or 10 pathways that are well known, sometimes called hallmarks, and things like the ends of your chromosomes, the telomeres get shorter, your mitochondria get dysfunctional, etc. And for most of those pathways, we have shown that we can reverse them. And there's maybe 8 genes that we and others have used in various gene therapies and combination therapies that reverse some of those pathways. But I don't think we've quite reversed all 10 of them at once. But we're very close. And like most things in biotech and electronics, is getting exponentially better.
Right. And like most things, at least in my experience in electronics, it's simultaneously closer than you think and farther than you might imagine. I guess it happens. Scientific progress does not happen in a continuum, as you mentioned. Right. It's discontinuous, and you never know.
But when it's discontinuous and exponential,
wow,
you don't have to wait long. And we didn't have to wait long to get from a completely unaffordable, inaccurate genome to a $300 clinically useful
Let's
one.
talk about that, because you founded a company a few years ago called Nebula that is different from so right now there are a couple of companies that do, they say, DNA analysis. I've done 23andMe and spit into a tube and so forth.
Right.
That's different from full genome sequencing. It's different from what Nebula does. Is that right?
That's right. There are a few companies that do full genome sequencing. Nebula is one of was the first that brought it down sort of in the $300 range.
Amazing.
Another company that I started, Veritas, was the first to bring it down to $1,000 range before that.
I applied for the Personal Genome Project, and I I wasn't accepted, or I don't know. Nothing ever happened, but I had read Esther Dyson did it. And one of the things that you were very clear about in the Personal Genome Project is, we're going to tell people about your genome. There's no way to keep this private. So we're not going to even make any assertion that there's any privacy here.
Right.
And I think for a lot of people, that probably would scare them off. When I told people that I had applied,
we wanted to scare off people.
Right.
Yeah.
When I told people I was going to do this, they said, "Well, you should get your permission from your children, because, in a way, you're giving away this critical information about your other people, not just yourself."
Yeah. Well, first of all, just because you're giving away your information in the Personal Genome Project doesn't mean that you have to give it away if you get a private genome, right? So the Personal Genome Project was to enable you to do that in a way that you're fully informed.
Right.
But you can also get one from Nebula, which is completely...
Well, that's what I'm curious about. It's changed.
Encrypted.
Yeah.
That's right. So we have ways that you can ask questions, you and your physician can ask questions of it in a completely encrypted form. It's got a jargon name of homomorphic encryption query. But the main thing is that you can keep your genome encrypted so that, if the government asks for it, you hand it over, but it's encrypted. But on the other hand, even if it were public, which we have now thousands that are public in the Personal Genome Project, you can't tell much about the children, because in principle, if you had like 20 children, you could tell something about the parents.
Yeah.
But if you have one parent or even two parents, you can't tell what any child has because of the random assortment. And so it's a very poor predictor. And even if you had the whole genome of the children, there's not that much you can tell about them. Especially if they've made it to a certain age, that means they've gotten past a lot of the most serious diseases.
Right.
But the point is not to reassure people about this. It's to maybe if they're worried, then they should go with the more secure version.
Yeah. When I applied for this, I don't think there was an option at that time. I mean, but I was perfectly.
There's still not really an option. Yeah. You can. Oh, you mean that? Yeah.
The privacy
it wasn't
option.
really a good way of getting a commercial private genome. Yes.
Right. Mm-hmm. But I would I was hurt that I was not accepted. I'm sure I'm sure there is.
It was probably a time where we were having trouble paying for all the genomes.
Yeah.
Because there was such enthusiasm for it.
Yeah. Exactly. It was expensive. It was, I think, $10,000 at the time.
When we started, we had brought it down to 350,000. So we actually sold through a third company that I started. The first of three was called Gnome, and that sold its genomes for $350,000 to fairly high net worth individuals, some of which became investors.
Yes.
And I started that about the same time that I helped Ann Wojcicki start
23andMe. So you were involved in that.
In 2007.
Yeah.
Mm-hmm. But they were very far apart in their business model. One was more focused on ancestry and kind of exploring common alleles, things that are so common in the population that both alleles, both variations that are harmless or normal, wild type.
Yeah, I mean, I got the impression that it was almost a statistical result rather than a personal result. Is that accurate?
No, it was statistical in the sense that you could take a common allele and infer what might be nearby that might be rarer and more disease oriented, but it wasn't really good enough. What you really need to do is sequence the causative variation. And that's what whole genome sequencing gets for you. I mean, we were only doing about a tiny fraction of 1% of the genome for 23andMe. But even Gnome was doing 90+%.
So now we can do
a lot more expensive. It was like, $99 or 350,000.
Yeah.
But now they're very close to the same price.
That's interesting. And and I think, I did it. My kids did it. My everybody in my family, my parents did it. I somewhat out of curiosity. You're right. It was about the genetic, the ancestry. But there was always that kind of interest in your medical history and information. Can we know now more? If I did Nebula, for instance, what kind of information would I get? What can my doctor do with that?
Well, the most useful thing is for people that are of reproductive age can find out if they're carriers of a serious disease, and either they can decide on who to date based on that information.
So you might want to say, hey, before I date you, you've got to do this so that we can make sure that we're okay.
Well, yeah. I mean, if you do it before you even meet a person, then you never really know what you're missing. You're missing at most 3% of the people that you would otherwise meet.
Right.
But practically, people wait until later when the longer you wait, the more and more critical it becomes.
Right.
So if you wait until you've already got pregnancy, then you're facing potential termination or either spontaneous abortion because they're so sick,
right.
or elective.
There's still somewhat of people do it somewhat out of curiosity as much as anything else, I would guess. Still, yeah.
That may be, but it really should be treated as a very cost-effective preventative medicine.
Okay. And are physicians aware of this? Are they increasingly using this kind of information?
It's a slow revolution. I mean, the technological part of it was fast, but I think the societal part,
right.
There will probably be some point where just suddenly everybody wakes up. I remember, there was a time where nobody wanted to use cell phones because they were afraid they were going to melt their head, and there was a time where nobody wanted to use seatbelts for similarly semi-rational reasons. And then it just shifts. I mean, sometimes it's government action where they put the Surgeon General has determined on the cigarette packs, and sometimes it's some influential personality who has a personal story that affects people. I don't know what it's going to be in this case, but
Right.
it will happen.
There is a reference genome, right, for the human genome.
There is. It's more like a mixture. Because you want to represent the diversity of the planet, and there are definitely certain populations that have thousands of new genes relative to another population. So the reference really has to represent some diversity. It doesn't have to. You don't necessarily have to have 8 billion people as part of the reference. That would kind of defeat the purpose of the reference. But you need more than one.
Is it safe to say there are 8 billion possible? I mean, everybody's genome is unique, right?
Yeah, even identical twins, even your left and right arm have some differences.
Interesting.
I mean, there's somatic variation. There's about somewhere between 10 and 100 changes per generation. There's a lot of variation, but most of it's neutral. It doesn't really impact your health.
And is there still a lot of the genome that we don't understand, or that I've heard it referred to as junk DNA? And
well, people use the term junk DNA less. I did my thesis on two regions that one might have thought were junk, and turned out I showed they weren't. But there's a lot that we don't know, and there's even a lot that we may not need to know. There's for example, some of the favorite organisms for engineering for industrial purposes and agricultural purposes. You can do a lot of amazing engineering only knowing a little bit. It's kind of like you can do a lot of engineering in mechanical terms with a limited part of the periodic table. Just got a long way with just iron and copper and
Right.
a few other things.
Well, let's talk a little bit about a project you've been working on to bring back the mastodon. Not exactly bring it back.
Slight nuance there is we're actually trying to make cold-resistant elephants, and we're using mammoth DNA to inspire us, not necessarily to compulsively go for although I think we will develop technology where we could do every single base pair change, but probably by the time we get to that, it'll be considered diminishing returns.
I'm not going to channel Jeff Goldblum and say you shouldn't do it just because you can. We're not close to Jurassic Park, I guess, is the. Although I wouldn't mind seeing a woolly mammoth wander around once in a while.
Yeah, well, hopefully we will have something that looks and acts and survives minus 40 winters, just like a woolly mammoth. But it may be a very small number of genes we have to change to achieve that.
And let's talk about the reason why you would want to bring back cold-resistant elephants.
So elephants are so-called keystone species in almost all the environments they've been in. They've been populated almost every continent of the planet at some point in history, and if we think of this as rewilding, so we're interested in endangered species and making especially the ones that have a strong positive impact on the environment, either greater diversity or something that humans want. In this case, it would be both. In the Arctic, the environment is much less diverse and is releasing a lot of methane due to the warming of the soil. Last time I was in Siberia, a couple of years ago, it was the first time in history that the summer thaw was not refrozen during the winter. So the Arctic in general is going up in its temperature about 4 times faster than most other parts of the planet. So anyway, that's releasing methane, and the elephants as a keystone species would enable other herbivores to maintain a grassland, which is what it was for many millennia. And that would result in better photosynthesis, better reflection of heat, and better conduction of the winter wind by allowing the herbivores to trample it. So there are reasons for endangered species. There are reasons for restoring the Arctic diversity, and the main one being that we can sequester carbon and keep it sequestered better.
Elephants knock down trees. I've seen you say they trample the grass.
Right.
You often think of insect species like bees as being important to the ecosystems, but a giant thing like an elephant, you feel like, well, how much impact could an elephant have? It obviously can have quite a bit of an impact in an area like that.
And there's precedent for this sort of thing of bringing back one keystone species in order to achieve great change. So in Yellowstone, the wolves have been gone for 70 years. People eliminated them, and then people brought them back. And when they brought them back, they intimidated the larger herbivores like deer and elk, which allowed the willows and other trees to finally grow. They had been gone for most of those 70 years. And then when they came back, the beavers started turning them into homes that would alter the create lakes and rivers, and hence would affect the waterfowl and the fish and the amphibian. It just ramified throughout the entire Yellowstone Park area in a way that was well documented. So this is one species that had such a positive impact.
Isn't that interesting?
And that's been repeated for maybe 1,000 different rewilding events worldwide since then.
My natural inclination is, God, these ecosystems are so complex. How can we know what levers to pull, and what consequences they might have? But I guess it's the same thing we say about gene splicing and everything that we're talking about.
And medicine. I mean, medicine. I guess the human body is incredibly complicated.
Right.
I think what happens is you need to do it in such a manner that you have thought through in advance how you might reverse it, how you might keep it contained. For example, in medicine, you might do a clinical trial that involves one or two people. And then if something goes wrong, that's a tragedy for one or two people, but at least you didn't introduce it to millions of people on day one. And the same thing goes for ecosystems. You can try it out on a small scale. Good. Especially if they're large animals. If you introduce a bunch of bacteria or mosquitoes or something, that's a little hard to reverse. But large animals, relatively easy.
You also invented from the macro, from the cold resistant elephant to the micro. You also have come up with a use of DNA for detecting dark matter.
Yeah, so I've had a number of pleasant interactions with physics colleagues, and that was one that some group years later used it for detecting other kinds of particles. It's still unknown exactly what dark matter is. There's a recent, I think, intriguing hypothesis for what dark energy is involving black holes, but I think we're still anyway. It was a general particle detector that was fun to. We didn't manufacture the detector, but it was in principle much cheaper than the other existing detectors at the time.
Is there anything genes can't do? Kind of amazing. If you could live forever, would you want to live forever?
Forever is a long time. I'd take it a little bit at a time and see how it goes.
A little longer.
It's what's hard to, people who say they want to live a normal lifetime. I just wonder whether most people are not willing to commit suicide. And if you're very healthy and youthful, I think it would be hard to say no to another couple of days, to say no to seeing your great-grandchildren and get married.
Yeah.
So I think it's naive to just casually say, oh, I'm going to willingly die at age 70, even if I feel like I'm 25.
Right.
I think that's a little hard to say.
No, that's a good point. and I'm open to any ideas you have. I mean, the funny thing is we all know already a great number of things we could do to improve quality of life and aging. Doesn't mean we do them. Be much nicer to take a pill.
Right. Yeah.
Yeah. One of the questions I wanted to ask is, I know Nebula, you use a Chinese company to do the sequencing. Is that correct?
Nebula is and Veritas have their own instruments.
Oh, okay.
And some of those are, most of them are from Illumina, but they're also from use Oxford Nanopore, which is a UK United States collaboration. Some of them are MGI, which started as CGI in California and is now merged with BGI in China. And Ultima is a new one that we're using as well. So it's a mixture. And I'm sure if a particular client wants to use only one technology, that can be arranged.
Given the recent concerns about Chinese spying and so forth, I think people might be a little concerned about saying, well, you're going to send my genetic.
I like to encourage people to be concerned. Some people think scientists are supposed to reassure us. I think scientists should be open to people's concerns and take them seriously. And I think that I would worry about spying by Californians as much as everybody else. I think that you need to. If you want it to be private, there are ways that you can build into the hardware encryption so that it's always encrypted. So there's never a decrypted form of it. And getting back to we can now ask questions of it, a limited number of questions, not enough to figure out the genome, but enough to answer your medical needs. So you could have it, cradle to grave encrypted. So if we want that, we could do that. But then the other thing we have to deal with is if people want your genome enough, all they have to do is follow you around because you're like spewing genome from every part of your body all day long. So the only way to really keep your genome safe, it's not who you choose to sequence your genome. It's more a matter of staying inside your house.
That's a good point. I didn't even think about it. Although I remember the movie Gattaca where he was trying to scrub all the dead skin off so that.
Yeah, he was trying to clean up his keyboard and stuff.
Yeah.
So, and that's real. That you can sequence a person's genome from one cell, and it can be a dead cell.
Right.
And we've sequenced DNA that's up to 2 million years old. So watch out if you're worried about that. But I don't think you really need to worry about it that much, at least not yet. I'm not trying to reassure anybody here. I'm just
no. I'm not reassured. Good news.
Good, yeah.
In fact, you just scared me even more because as it becomes cheaper and cheaper and more available to sequence, I'm going to start keeping my nail clippings and my hairbrush away from the people because it's all there.
And your coffee cups and for your dandruff, et cetera. So, Head and Shoulders is going to make a tidy profit.
Really, even from dandruff, we have recreated a couple of germ sequences I thought were kind of interesting. One was the 1918 flu from a fella who was frozen in the earth.
Yeah. We can also reconstruct old things that are, found in our genome. We reconstructed an ancient retrovirus from the human that's present in everybody's genome, but it had decayed a little bit. You can reconstruct that. Yeah.
Also risky. Yeah.
Yeah. And they've also made gain of function viruses, so things that don't exist in nature that are worse than pathogens in nature. And they did that without general consensus on it in advance. It was basically they submitted it to journal. It was the first time that anybody outside their lab knew about it.
Right.
And that's probably, I think, with hindsight, and maybe some people would have said with foresight, that's not a good idea. And it's
that's what the Wuhan lab in China was engaged in. Yeah.
That was
Weren't they doing gain of function research?
Well, they've done gain of function in almost every major country. I think the the gain of function for flu, there was one for flu and pox virus. Those were done in the United States, England, and Australia.
Yeah. And there's reason to do that. Just as there's reason to resurrect the 1918 flu virus. Yes,
there is. It can be justified in that we want to get ahead of these things before they appear. It's not. And it's hard to say, oh, we're going to figure it out and then keep it secret, because then somehow it'll get into the wrong hands, and then only the people with the wrong hands have it. So if you're going to do it, probably make it public. Anyway, arguments can be made on both sides. I personally don't do that research, but I'm not necessarily judging people who do.
And speaking of hands, I have to ask you about the doomsday clock, because you're now on the board of the Bulletin of Atomic Scientists. I see. Right. Do you get to vote on the status of the doomsday clock?
I think yes, but I think my vote has about as much sway as my vote in the last presidential election.
There are a lot of you, eh?
There's a lot of people voting on that clock, and it seems like there's some kind of market forces that keep the clock very close to midnight. It's more exciting when or more depressing when it's close to midnight, more alarming, and we deserve to be alarmed, quite frankly. I mean, there's all sorts of things we're doing in terms of new forms of warfare, testing them out on Ukrainians. New, cyber warfare, bio warfare, and all these things are unnecessary, I think, to maintain. In fact, they're contrary to maintaining a high standard of living. Russia almost went broke competing with America on the Cold War.
Right.
And I think all of these things have economic consequences. It would be better if everybody worked on eliminating diseases of poverty, working on better educational systems and so forth. Then we would have a virtuous cycle rather than a vicious cycle.
You strike me as an optimist. Are you an optimist?
I don't think it's that simple. I think I can see a pathway, but I am pessimistic in that I don't see people following that pathway. So I think eventually they do. It's just
will they do it in time is the question, I guess.
Yeah, exactly. I mean, they have. I think I should include myself a little bit here. We humans have a tendency to ignore things that have a risk of less than 1%.
Yeah.
Maybe even for some people less than 10%. So you say, 90% chance of winning, I'm going to Vegas, right? Makes sense when you frame it that way. When you say, well, a 10% chance of dying if I keep doing what I'm doing, that doesn't sound good, especially if it's 10% compounded daily or annually or something like that. And that means you're pretty much for sure going to screw up the planet. So I think maybe we should just drop all education except for risk assessment. I mean,
it'd be valuable to understand.
A lot of things we learn in school we never use in our profession,
right?
And one of the major things we don't learn in school apparently is how to do risk assessment.
Right. We're terrible with probability. Completely enumerate.
It's mostly low probabilities applied to our personal decisions.
Right.
We would prefer to take a risk. It's kind of like, well, look, the heroes in World War II, they took risks. Why shouldn't we? It's like, why should we if we have an alternative? I think the heroes in World War II really had no alternative.
Right. What is it? What's going on right now that you're most excited about, that you're working on?
We've touched on a lot of them. Certainly, aging reversal would be handy for getting me a few more years.
As we get into our 60s,
research.
George, we're both very interested in this.
I'm 68, but it's not just selfish. I think a number of people that have been educated for 60 years that's a precious resource, and we shouldn't just throw it out because it would be like spending 60 years building a supercomputer and programming it and so forth, and then the day that you would turn it on, you recycle it,
right?
And turn it into soda cans and stuff. That just doesn't make sense to me,
right? So actually, that's the argument for. I mean, from a purely biological point of view, there's no reason for us to exist once we've reproduced. But there's the argument for it. We've got
but except a huge fraction of our evolution has enhanced our ability to live beyond reproduction.
Right.
I mean, even as cave dwellers, we had value added by the grandparents, and in some cases even great grandparents that existed long before advanced medicine, and they're the ones that remember that floods happen every 50 years, and you have to prepare for droughts and famines and so forth, and it's that memory. You can see that wars happen when all the people that suffered from the wars die off. Then everybody thinks, oh, it was very glorious, and let's do another one. So I think there's some benefit there. But you asked what I was excited about. Another thing I'm excited about. I mean, almost anything has really kind of basic science and societal benefit gets me excited. And one that we recently just published in preprint form, and hopefully it'll be out. It's been accepted at a major journal, peer review. Is we found a way to make an organism resistant to all viruses, including viruses we've never seen before. And we proved this by going out and collecting sewage samples and farm samples that had completely unknown viruses in them, and showed that it was resistant not only to all the laboratory strains, but all these wild strains as well. And we did it by a method I think is completely general. We can now make any organism, in principle, resistant to all viruses by just swapping two amino acids, serine and leucine, at a particular place at multiple places throughout the genome.
Wow, that sounds pretty exciting.
Yeah, you asked me what I'm excited about.
That's exciting.
That's an example. That's an example.
Yeah.
It's not the only example. There are a few others. But that's you work on something for a while and then it finally arrives.
That's amazing. Yeah. Well, it's been a great honor and a privilege to talk to you, Dr. Church. The work you're doing, the work you've done, transformational, and
thank you.
A grateful country thanks you. We really appreciate it.
It's sufficient just to see these things used in medicine and agriculture, and maybe even ecosystems soon.
Yeah. I wouldn't mind seeing a mastodon once in a while. I think it'd be kind of cool. Thank you, Dr. Church. I appreciate you joining us. Have a great day.
My pleasure. Thank you very much. You.
What a thrill to talk to Professor George Church. I should mention that Nebula, his genome sequencing company, is open to all at nebula.org. If you're interested. His book, Regenesis, is still available on Amazon. He wrote it a few years ago. There are a number of TED Talks featuring Dr. Church if you want to know more about the woolly mammoths and some of his other projects. But we're so glad we were able to get him for an hour today on Triangulation. Thanks for joining us. As you know, Triangulation is not an every week event. It's whenever I can find somebody that I really want to talk to. We have been doing a lot more lately, and I hope we will do more. One of the reasons we can is because of Club Twit. So I want to thank our Club Twit members and remind you that if you're not a member, your $7 a month can make such a difference, keeping us on the air, keeping our employees hired, and doing shows like this, shows without advertising because of our Club Twit members. You don't just get all of our shows ad-free. You get shows we don't put out any other place, like Hands on Macintosh, Hands on Windows, the Untitled Linux Show, the Giz Fizz. You also get access to a really great community in our Club Twit Discord. Thousands of people in there, all like you, with an interest in technology and geek subjects, everything from anime to cooking to travel to beer. If you're not a member, please consider joining. We really appreciate it. twit.tv/Club Twit. And I thank our Club Twit members for making this possible. I thank you for joining me. We'll see you next time on Triangulation. Bye-bye.
Do you want to hear about the latest news happening in the tech world from the people who write the articles, sometimes from the people who are actually making the news? Well, we got a show for you here at twit.tv. It's called Tech News Weekly. Me, Jason Howell, and my co-host Micah Sargent, we talk with some amazing people each and every Thursday on Tech News Weekly, and we share a little bit of our own insights in each of us bringing a story of the week. That's at twit.tv/TNW. Subscribe right now.